Good morning, good afternoon, good evening, everyone. It's very early here in the northeast of the US. Today, the task of the day for me is to introduce the last but not least keynote presentation of the meeting. It is a pleasure for me to introduce Francois Marais. Although Francois is currently the Director of Biologics and Infectious Diseases section at Clean Global, his career for the past 20 years has been dedicated to working in the F&D field. He started his adventure when he joined the Agricultural Research Council in Pretoria, South Africa in 2003. And since then, he has become a key player in the studies of FMDV in the South African territories SAT serotypes. He was the first to generate an infectious clone for an FMDV SAT serotype, and that opened the path for his research later on. His group has been focused on the improvement of FMD vaccines using recombinant DNA technologies and reverse genetics, with several contributions towards the custom engineer and structural design of improved FMD vaccines. By improving virus yield in infected cell culture, virus capsid stability, identification of viral epitopes, and source of antigenic variation. Another very important aspect of his research focuses on the pathogenesis, transmission, and persistence studies in African buffalo. In fact, this is the topic of his presentation today. So with this, I pass it to you, Francois. The floor is yours.
Good morning to all. I would like to thank the organizers of the GFRA for the opportunity to present the work done on the transmission of SAT2 viruses among buffalo and the pathogenesis of SAT2 in cattle. The work was done while I was at the ARC, but it's an honor to present it in my new capacity at Clean Global. In Southern Africa, buffalo are considered the natural reservoir of SAT1, SAT2, and SAT3 foot-and-mouth disease viruses. Serological surveys have shown that by the age of 2, majority of buffalo have been exposed to these three serotypes. Buffalo are thus efficient FMDV carriers and are the source for the generation of new genetic and antigenic variants. Two epidemiological cycles of foot-and-mouth disease are recognized in Southern Africa. One, an endemic cycle involving buffalo persistently infected with SAT viruses, and two, escape either directly from buffalo or through the involvement of other cloven-hoofed wildlife to susceptible livestock, followed by a cycle primarily involving livestock. Mapping cattle densities and buffalo herds in Africa, Casa et al. Showed an overlap of buffalo and cattle population, especially in Southern Africa, and this can also clearly be seen in the area neighboring the Kruger National Park in the southeastern corner of South Africa. Although there are few experimental reports supporting buffalo to cattle transmission, there is plenty of phylogenetic evidence in Southern Africa that demonstrates that the transmission from buffalo to FMD-free cattle does occur. A high proportion of foot-and-mouth disease outbreaks in Southern Africa is caused by SAT2 viruses. Now, SAT viruses have been reported to cause a range of mild or subclinical infections that sometimes may go undiagnosed in field conditions. So we report here on the pathogenesis and the range of pathogenesis of SAT2 viruses, and also the relative infectivity and virus fitness of SAT2 viruses in cell culture. And this is work that has been published by Ramlonga et al. SAT viruses, as I've mentioned, are maintained effectively in African buffalo. However, the mechanisms of persistence remain unclear. And the data that we present here come from 3 different studies. First of all, an outbreak investigation on a farm, experimental transmission studies in the Kruger National Park, and then also ecological study where we follow transmission dynamics within a cohort in the Kruger National Park. For the pathogenesis work, we selected 4 viruses. ZIM 1490 and ZIM 583 originated from cattle outbreaks in different regions in southern Africa. The ZIM 783 virus originates from ZIM 583 through multiple rounds of passages through BHK cells and cattle to generate a virus with a passage history of at least 16. The 4th virus was a sequence of a virus causing an outbreak in Egypt in 2012, cloned into an infectious genome length clone called SAT2 to generate the V-Egypt virus. The ZIM5 and ZIM783 infectious material from an initial challenge was collected and used for pathogenesis study to compare with ZIM1419 in vivo challenge. The Egypt pathogenesis in cattle have been reported in literature before, and it has been reported to cause severe lesions and myocarditis in young animals. Cattle inoculated with either ZIM5 or ZIM783 showed mild symptoms with only 1 or 2 lesions to sites other than the inoculation site and mild fever. Now, viremia was detected around 2 to 4 days post-inoculation, but viral RNA started to decline when antibodies started to increase from around 5 days post-infection. Now, this pathogenesis compared to that of ZIM1490, where ZIM1490 caused clinical scores of 9 to 12, which were observed from 2 to 4 days post-infection. However, lesions appeared as early as 1 day post-infection and spread to all 4 limbs in the case of ZIM1490 inoculated cattle. Viral RNA and in blood was generally higher than that for ZIM5 and ZIM7, and antibody titers started to increase at an earlier stage. Plaque morphologies on BHK and ZZR for these viruses were comparable. However, the Egypt virus produced needle head-sized plaques after 48 hours infection. ZIM7 and the VSAT2 and Egypt viruses were able to grow on CHO cells, implying that these viruses use receptors other than the integrin receptors generally used for cell entry. Growth kinetics of ZIM viruses on the BHK cells were comparable, although ZIM783 provided higher titers at the various time points of collection. The growth of the Egypt virus were also similar in the initial phases of growth. However, viral titers were considerably lower than the other viruses at the various collection time point. The cell killing assay comparing ZIM7 and ZIM5 were comparable, and even though they have different characteristics on some of the other measures that we use, they killed cells in the same manner. Infection of BHK and ZZL cells with the VSAT2 originating from ZIM783 and the Egypt virus individually both produced similar number of RNA copies across 10 passages. However, when these viruses were used to co-infect these 2 cell types, the Egypt virus was replaced by the VSAT2, and after passage 5, the viral RNA of the Egypt virus started to disappear. By passage 10, no more Egypt virus RNA could be detected. In summary, the SAT2 viruses cause mild to severe clinical or subclinical infection in cattle, and it may go undetected in field conditions. Therefore, this emphasized the need of heightened surveillance in regions where SAT2 infections are prevalent. Replication dynamics and fitness in cultured cells was related to the number of passages and not so much to the pathogenesis of SAT2 in cattle. Next, I'd like to share some observations made during an outbreak investigation on a buffalo farm called Bushwillow Creek. Bushwillow Creek is situated in an FMD-free region of South Africa. The farm contained 3 herds of buffalo in 3 separate camps, 2 breeding camps and a hunting camp, all FMD-free. The farm is surrounded by citrus farms with no cattle or buffalo in the vicinity. No other cloven-hoofed animals have been detected with any clinical lesions or signs in the past. The scenario is that Bushwillow Creek has 2 breeding camps and 1 hunting camp. The hunting camp contained about 40 bulls, and in September 2018, about half of these bulls were moved to a boma, and 2 months later, 4 females from the breeding camps were also moved to the bomas to be sold. So in September 2018, these bulls, about 11 of them were tested, blood were collected, and they were found, 9 were found to be positive for antibodies against SAT2. So for the first time, antibodies to FMD have been detected on this farm. And it can be concluded that these animals were exposed. How did it happen? When did it happen is not known. Was there an outbreak at this point of time, or did the outbreak happen at the previous time? The same animals were tested again in December and also tested positive for antibodies to SAT2. In 2019, we started with an extensive sampling. We selected again 11 animals, 7 from the bulls that was positive before, and the 4 females that were moved into the bomas. Of the 7 bulls, 5 of them tested positive again for antibodies to SAT2. However, 2 that was negative in the previous round stayed negative. And also the females from the breeding camp were negative. It's important to note that no virus could be isolated or nucleic acid detected from any of the proband or tonsil swab samples that was collected. So it's only antibodies. Again, we don't know whether the outbreak happened at this point of time or at an earlier point. We followed up with another sampling about 4 weeks later in March 2019, and again, the bulls that was positive originally stayed positive. And then the 2 negative bulls were negative, the females were negative, and there were 2 others that was negative as well. This one here, which is called Madala, and then the bull B53. Now, let's look at the movements that happened during the last 3 years before the detection of SAT2 antibodies. So, Madala was a dominant bull in the breeding camp. He was pushed out at some point by a newcomer called Rolf. Madala escaped into the hunting camp by himself and stayed solitary all the time. At some point around August 2017, Madala has been moved to a separate breeding camp, and 4 females were put into that breeding camp with Madala as well. They all still stayed seronegative. Then in December 2016, 2 young bulls called Dumpling and Knopkop that was born in this breeding camp and grew up there were moved to the hunting camp. But soon after, Dumpling found its way back to the breeding camp. Then in April 2017, Dumpling and another young bull, Big Boss, was moved to the hunting camp again. Knopkop seroconverted and had antibodies to SAT2. However, Dumpling and Big Boss stayed negative. In August 2017, a bull named B53 was also moved to the hunting camp, and that bull was also negative. So again, we do not know when exactly this outbreak took place, but clearly from this data, there is not an active infection and transmission any longer. So let's look at some of the studies that we have done to see if we can shed some light on the scenario there. During the first challenge study, we inoculated 16 buffalo with a SAT1, SAT2, and SAT3 virus each, and follow this virus persistence over time. Now, the first for buffalo was terminated about 30 days after the challenge to determine whether persistent infection took place. And although Nguni cattle developed extensive lesions, we didn't saw any lesions other than the inoculation sites at any of these buffalo that developed. Also, we detected viremia, and all buffalo seroconverted to all serotypes about 6 to 11 days post-infection. With virus isolations from day 30 onwards till the end of the experiment 12 months later, showed that virus could be isolated at any of these time points from most of these viruses. SAT1 was isolated most of the time and for a much longer period as well. Detection of viral RNA in tonsil swabs and probing samples supported the virus isolation results. The proportion of samples that stayed positive for RNA of each serotype decreased over time. However, this rate of decrease in the viral RNA was different for each serotype. And strikingly, the SAT2 RNA disappeared quicker. Roughly around 100, after 172 days, we couldn't detect any SAT2 viral RNA anymore. While SAT1 RNA was still detectable at 12 months after challenge in both these sample types. So in summary, we observed less transmission of SAT2 from carriers in this experimental challenge buffalo. SAT2 persisted for a shorter time period in buffalo compared to SAT1 and SAT3, and significant number of animals in this experiment failed to maintain a persistent SAT2 infection. Also, it appears that SAT2 got displaced by SAT1 and SAT3 during co-infection in buffalo. We also confirmed it in our paper, Maria et al. 2016, in co-infection in cultured cells. So, there was no live virus isolated at the time of sampling at Bushwillow Creek for phylogenetic comparison. It implies that the outbreak scenario most likely happened at an earlier stage. So we still don't know what the duration of SAT2 infection in buffalo is. What are the transmission dynamics of SAT viruses in buffalo? And how persistent is SAT2 virus infections and possible transmission from carrier states? So in a consortium consisting of all members of all these organizations and published recently in Science by Jones et al., we did a study on the endemic persistence of foot-and-mouth disease in buffalo. And there are especially two aspects to the study that I would like to highlight, and that is a cohort sampling in an isolated buffalo herd within the Kruger National Park, as well as a challenge study. During the first part of the study, a herd of buffalo in a fence camp in Kruger National Park were followed and sampled every 2 to 4 months for a period of 3 years. From the seroprevalence data, it is clear that SAT1 gives a robust antibody response that lasted for a long time and was stable over time. SAT3 antibody responses generally were short-lived and disappeared quickly and appeared again. So that said, it is important to note from this data that buffalo frequently lose their protective titers and frequently change from seronegative to seropositive. Although this change was not linked to age, sex, and nutrition. Also, no primary SAT2 infections were detected in these buffalo. During the second part of the study, 2 buffalo challenged with each serotype were mixed with 6 naive buffalo, and the transmission events between persistent infected buffalo and the naive ones were monitored at 4-week intervals. 2 identical groups were created in this manner to study the transmission with or without stresses. Now, transmission took place in the absence of stresses. and were detected within the first 30 days of contact between the naive and persistently infected animals. Transmissions of SAT1 viruses were observed first. However, no transmission of SAT2 was observed during the persistent phase, as we can see from the RNA copy numbers that was generated from real-time PCR. Over time, SAT2 and SAT3 viral RNA also disappeared and was most likely outcompeted by SAT1, something that we saw in cell cultures as well. The modeler team of Anna Jules used this information from both the cohort and the challenge study and determined that following primary infections with the different SAT viruses, the estimated proportion of buffalo that become carriers are very different for the SAT viruses. Also, the duration of the carrier status was quite different. But strikingly, the transmission rate from carrier buffalo were very different, and it was especially observed with SAT2. Now, using the predicted transmission from the carrier animals, SAT2 fails to persist in these models. And the reason for that is the low estimated carrier transmission rate. Now, coming back to the Bush Willow Creek scenario, the fact that there's a low probability of persistence of SAT2 in buffalo populations and transmissions from these carrier animals could explain the scenario there. And the ideas that I would like to leave with you is that SAT1 but also SAT3 viruses were transmitted from carrier buffalo when in contact with naive buffalo. Transmission occurred in the absence of obvious triggers, for example, protein restriction diet or co-infection with respiratory pathogens. There was different transmission rates observed for the different SAT viruses. SAT1 viruses transmitted relatively easy from carrier buffalo, and this allowed SAT1 virus strains to persist within small buffalo herds. SAT3 viruses transmitted intermittently, and long-term persistence is possible with larger herds of around 2,000 or more. However, the probability of persistence of SAT2 viruses in buffalo are low. And the duration of carrier status is also much shorter than with the other SAT viruses. So in the cohort study, we also found no primary or chronic SAT2 infections, which suggested that SAT2 may have gone extinct in the buffalo herd. And these experimental observations supported the observation at Bush Willow Creek. I just want to like to thank all the collaborators within the consortium that made this work possible. It was an honor to work with all these people. Thank you. I'd like to thank all the collaborators within the consortium.
Thank you so much, Francois. Great presentation. We have some questions in the chat from Giselle Medina. Is there any evidence that the viral determinants in the SAT viruses may be influencing persistent infection in buffaloes? Thank you so much, Francois. Great presentation. Have some questions in the chat from Giselle Medina. Is there any evidence that the viral determinants that viruses may be influencing persistent infection?
Okay, thank you for the question. It's a really good question. With the sequences that we have available for the viruses that we use as challenge material, there was no clear link between sequences and the virulence or pathogenesis in buffalo or the transmission in buffalo. So I think there's still a huge gap in our knowledge of sequence or factors, viral factors that may be involved in transmission in buffalo or pathogenesis in cattle even. So thank you.
Next question comes from Wilna. Can one extrapolate these results to all viruses within a serotype, or can one expect other strains within each serotype to behave differently? Next question comes from...
Thank you, Wilna. Yes, my opinion is that these characteristics is isolate dependent and not serotype dependent. That is, each serotype, or each isolate within a serotype may react differently in terms of its pathogenesis in cattle or its transmission in buffalo. One of the things that we really would like to do is actually follow up some of the buffalo challenge work with different SAT2 strains and even combination of SAT2 strains to confirm what we've seen with this specific SAT2 and the disappearance in buffalo with the SAT2 virus that we selected here is true over a wider range of viruses.
Perfect. Next question is from Don King. Can I ask an obvious question? He says, do you think that these observed effects are specific to the isolates, or are these genetic features of these serotypes? Next question is from Don. Can I ask an obvious question? Do you think that these observed effects are specific to the isolates, or are these genetic features of these serotypes?
It's not specific to an isolate. Sorry, the wrong way around. It's not specific to a serotype, but rather specific to an isolate.
Perfect. Next one is from Toby Tuthill. Two of the isolates in your pathogenicity study grew on CHO cells. Do these strains have sequence consistent with HS or fivefold adaptation? Could this be a problem of pathogenesis in these strains?
Thank you, Toby. Well, so the three strains that we put through cattle and look at the pathogenesis, the ZIM 1419 and ZIM 583, they did not grow on CHO cells. But ZIM 783, which is a derivative of ZIM 583, did grow on CHO cells. The pathogenesis of ZIM 5 and ZIM 7 were mild versus ZIM 1490, which were severe. So there wasn't a clear link between the ability to use heparan sulfate and the pathogenesis in cattle.
Perfect. Next one comes from Hydra Eble. Can you be sure that the isolates that you used in your study are representative for the whole serotype group?
No, definitely not. And that's why I said in a previous answer that we really would like to repeat these two challenges in buffalo. I still believe there is each isolate will behave somewhat differently, although there will be a spectrum of behavior within a serotype. So I don't think they necessarily representative.
Perfect. Next question is from Aldo Decker. The experimental work indicates that the SAT2 would die out in small populations, but he thinks, or he thought, that the SAT2 was the most prevalent serotype. How can you explain this?
Thank you, Aldo. Well, these experiments show that the SAT2 will die out in small population of buffalo. And the most prevalent of the three SATs causing outbreaks in cattle is SAT2. So
the behavior of these SATs in cattle it's not necessarily a linear extrapolation from buffalo.
Yeah, we have two other similar questions from Toby Tuchil and Frank Wanney. How would you explain displacement of the SAT2 and SAT3 by SAT1 viruses over time?
Frank, that is a very, very interesting question. That is also something that we think of a lot. In my opinion, it could be the adaptation of SAT1 to the specific species that we investigated, and that is buffalo. There seemed to be definitely a growth advantage in terms of persistence within buffalo. That's all I can say at this point. I don't think we have real answers for that.
Perfect, thank you. I think we have covered our time for this keynote presentation. Thank you so much, Francois, for your time. It was a great, great presentation here. Reminder for everybody, if you want to keep asking questions, you can find Francois in the Gather Town later during the break. With this, I would like to conclude this session and move over to the next session in epidemiology. Thank you very much.
Thank you, everyone.